We're going to talk about transcription, translation, and DNA replication in a short, quick, down and dirty way. First, let's look at some facts about DNA. DNA is a double helix, which means it has two strands that happen to be anti-parallel, meaning one starts at 3' and runs to a 5', so it starts with that hydroxyl group and goes to the phosphate, and the other one has the phosphate that goes to the hydroxyl group. So they kind of are they're anti-parallel, so they are opposite of each other. It's also a semi-conservative replication that happens. So when DNA replicates before mitosis, so before the cell splits, it replicates one side and it replicates the other side, and then it's semi-conservative, meaning that this strand right here, half of the new strand will be, or half of the replicated strand will be original DNA and the other will be new synthesized DNA, and the other strand of DNA will be half original DNA and half newly synthesized DNA. It's also made up of multiple nucleotides, and a nucleotide is a sugar, a base, and a phosphate. And so that sugar and base make up the backbone or the handles of the ladder, and then the bases attached together by hydrogen bonds make up the rungs of the ladder as if you were climbing up. When talking about DNA replication, there's a few things that happen. First off, the DNA gyrase comes along and makes, let's say we're going to synthesize right here, DNA gyrase. That enzyme comes and makes a little nick, and then let's see, it's going to color here. The topoisomerase kind of comes in and it will hold. Basically what it does is it unwinds the DNA so that it can be, so this comes in, it'd be right here, and it holds that, it kind of straightens out the DNA so that it can come through. So the helicase, so this is, let's see, right here is the topoisomerase. And that, like I said, straightens it out, and then you have the helicase, which is like the zipper. And this unzips the DNA from itself, so it essentially goes through and breaks those hydrogen bonds that are holding those base pairs together in the middle of the ladder. And then this replication fork, because the helicase comes in, it creates kind of like a little fork where the DNA can be replicated. And because, like we said earlier, it replicates from 5' to 3'. So one of these, and these are anti-parallel, so one of these is going to be a 5', and one of them is going to be a 3'. And the reason I know which one is which is because new DNA synthesizes from 5' to 3'. So this is known as the leading strand, because it is a continuous synthesis, and this is the lagging strand because it has to go from 5' to 3', but it can only do that in short bursts. These are also known as Okazaki fragments. That will later be attached by the DNA polymerase. So when the helicase unzips the DNA strands, there are single-stranded binding proteins that essentially hold this DNA open, because there are multiple bases that are attracted to each other, obviously, so they want to go back together, but these binding proteins help hold it apart, while the DNA primase kind of comes through, and the DNA primase is the first one that will read. It'll read the base. So this is a DNA. So these are binding proteins. And then this is DNA primase, like a primer. Helps get it started. So it'll read the bases, and then that primer will bring in the appropriate base. So if this is an A, what's it going to bring? It's going to go and find a T. And then it's going to bring that in right here. So then this little guy is a RNA primer. And each one of those, it's kind of an individual. So then you have DNA ligase. Let's get a different color. So DNA ligase comes in and seals up all the little pieces together. So it seals up all the new backbone together. So this is the DNA ligase. And then all the thing that zips the whole piece back up into a one big new strand of DNA is known as the DNA polymerase. So let's try it. So then this, and it'll come through and connect all these Okazaki fragments as well. This is the DNA polymerase. And it's like the zipper. Okay, so it goes through and it zips them up, and after the DNA polymerase matches up all the bases with hydrogen bonds, the DNA ligase has gone through and matched all the backbone pieces together, so those nucleotides that are being brought in by that RNA primer, so that the phosphate, the sugar, and the new base, after those have all been zipped up, the DNA winds itself back up, and you now have two new strands of DNA. Again, it's semi-conservative, so obviously, like you can see on this right here, this light blue and dark blue strands, these are going to be conserved. The light blue strand is going to be on this top strand, and the dark blue strand is going to be on this bottom strand. Transcription is very similar to DNA replication. So what we're doing in DNA replication is we are taking a copy of the DNA that we already have and making a whole new copy of all of the DNA, so that when the cell goes through mitosis, or that cell division, we'll have two identical copies of our DNA within both of those cells. Transcription is just taking a piece of DNA and reading it into an mRNA strand, and then that mRNA goes and codes for specific proteins. So similar to DNA replication, there is a nick that is made in the DNA, but instead of coding for both ends at the same time, what's happening is only one piece is used. So for example, we have this section right here that's being read for the mRNA strand. So with DNA, we have 4 bases. We have A, T, G, and C. And when we're talking about RNA, we only have 4 bases as well. However, thymine gets replaced with uracil, so it's A, U, G, and C. So if, for example, I have a code of DNA, so here's my DNA strand right here, let's say, and the code is G, C, A, T, T, A, G, C, my corresponding RNA code is going to match as well, but we're going to replace thymine with uracil. So it's going to be G pairs up with C, C pairs up with G, A pairs up with T in DNA, but if it's RNA, it's going to pair up with U. T pairs with A, T pairs with A pairs with U, G pairs with C, and C pairs with G. And then that piece of mRNA is going to leave the nucleus. So here's that piece of mRNA. It's going to leave the nucleus, and it's going to go into the cytoplasm and be read by that ribosome to create that protein. So then we have translation. So translation is taking that mRNA code, or those base pairing codes, and changing it into a functioning protein. So a functioning protein essentially is the gene expression. So it's what we actually see. It's that genotype that gets transformed into the phenotype that we actually physically see. So we have this piece right here is our little code. And so that basically tells us which amino acids or peptides are coded for by that codon. And so that codon is that 3 base pair. So let's say we have this 3 base pair and it is AUG. That is our start codon. So our start codon also codes, so AUG, that codes for MET. So that is the peptide that is going to be brought in to start that protein. So we have rRNA that helps link them together. So a ribosome reads the big long strand of mRNA. And at each codon, it's going to tell this tRNA right here, this is just a big strand of RNA. So this is tRNA. And that tRNA goes and gets that peptide or that piece of amino acid that is coded for by that mRNA strand. Then you have another one that comes in, another tRNA that reads, so that ribosome switches over, and it reads this codon, and then it switches over and reads another codon. And the tRNA, so all of these guys right here, are the tRNA bringing in the peptides. So here is an amino acid, here's an amino acid, and I use peptide and amino acid interchangeably, so it's the same thing, it's just a different word for it. So then I have my rRNA, or my ribosomal RNA, that links those peptides together. So if I have one peptide, it's a peptide. If I have 2 peptides, it's a dipeptide. 3 peptides is a tripeptide, and so on. Obviously, when you have many peptides, it's called a polypeptide. Poly meaning many. So this is a polypeptide chain. And essentially what that means is we are taking that multiple amino acid chain and forming it into a protein. So that polypeptide chain essentially winds up into a specific protein. And as you can see here, this is a wound up piece of protein. So it goes from the DNA, which is what essentially is our blueprint, right, for all of the genes that we have. This mRNA that made a copy, right, of just a portion of our DNA, this mRNA codes for a specific trait. So it goes in and it reads a specific gene. So for example, this gene, let's say, is blue eyes. So it's going to be that genotype, what your genes are telling you. So all those base pairs chemically tell you that you're going to have a certain eye color, a certain hair color, or a certain color of skin. And so this is the specific copy, right? So this blueprint might be for the entire house. This specific piece this is how this bedroom is going to look, and then this protein is what it looks like. So this is like the phenotype. Okay, or the physical trait. So this would be when the protein actually winds it up onto itself, it actually codes for this is what it actually looks like. So this, like I said, if the DNA is like the house, blueprints, right? And then you have a specific copy, so this is what your bedroom would look like. And this is what your actual bedroom looks like. So the actual of your bedroom. Hope that helps.